And welcome to the oro-oto-cerebral pathway. In the embryology series today, we are going to talk about the placenta. To understand the concept better, let us first revisit the background. We have seen that the trophoblast in the early second week helps in implantation. At that time, the trophoblast is subdivided into two layers, the syncytial trophoblast and the cytotrophoblast. Syncytial trophoblast is a multinucleated protoplasmic mass that is capable of attaching and eating up whatever structure or epithelial surface it comes in contact with. And this very property of the syncytial trophoblast actually successfully embeds the embryo into the wall of the uterus. When the embryo has gradually buried into the endometrium, spaces start appearing within the syncytial trophoblast. Those spaces eventually give rise to large spaces, and that is lacunar stage, so to say. As the lacunar stage progresses further, the syncytial trophoblast successfully erodes the maternal blood vessels, and now through the lacunae of the syncytial trophoblast, maternal blood starts flowing. This is said to be establishment of utero-placental circulation. The development of trophoblast doesn't stop there. After the formation of utero-placental circulation, the cytotrophoblast, which is the inner layer, starts sending finger-like projections through the syncytial trophoblast, and those finger-like projections are the chorionic villi. The chorionic villi go through three stages of development. There is a primary villus, there is a secondary villus, and then there is a tertiary villus. Primary villi consist of syncytial trophoblast and cytotrophoblast. The inner core is formed by cytotrophoblast, the outer cover by the syncytial trophoblast. This is day 13 approximately. By day 16, we see a secondary villus. In the secondary villus, the core is now occupied by the extra-embryonic mesoderm. In a span of next 4 or 5 days, say by day 21, the secondary villus has converted itself into a tertiary villus. The tertiary villi now have in their core blood vessels arising from the extra-embryonic mesoderm. At the level of tertiary villus, the cytotrophoblast kind of forces through the core of the villus in which it's already present and grows beyond the limits in order to form an outer shell around the entire villus network, and this outer shell is the cytotrophoblastic shell. So how does the division of labor work for this part? We have two entities, cytotrophoblast and syncytial trophoblast. Cytotrophoblast acts as the supportive framework. The cells divide and produce syncytial trophoblast cells. The syncytial trophoblast, on the other hand, produces hormones like human chorionic gonadotropin, the HCG, which is there to maintain the corpus luteum. It also secretes progesterone as well as estrogen. And to top it all, it also produces somatomamotropin, which modifies the metabolic status of the mother to facilitate the energy supply to the developing fetus. At the tertiary villus stage, the capillaries in the mesoderm of the villi come in contact with capillaries in the chorionic plate and the connecting stalk, and thus there is establishment of an extra-embryonic vascular system. By the beginning of the 4th month, cytotrophoblastic cells and some connective tissue cells of the villi disappear. Syncytium and endothelium of the fetal capillaries are the only layers that are left to separate the maternal and the fetal circulation. This is the blood placental barrier. Let us see more about it. The placental barrier or placental membrane consists of endothelium of fetal capillaries and their basement membrane. Secondly, the surrounding extra-embryonic mesoderm. Thirdly, cytotrophoblast and its basement membrane. And fourthly, syncytial trophoblast. This assembly of 4 layers amounts to about 20 to 30 micrometer in thickness, and this placental barrier is not going to stay for the rest of the pregnancy as the same 4 layers. By the time the pregnancy advances and we are witnessing advancement of pregnancy towards the late second and the third trimester, this barrier of 20 to 30 micrometer is going to thin to almost 2 to 5 micrometer, where instead of 4 we get only 2 layers, that is the syncytial trophoblast and the capillary endothelium. There is no mixing of fetal and maternal blood in the normal settings. As pregnancy advances, villi at the maternal pole continue to grow and expand, and this gives this area of chorion a bushy appearance, which is called chorion frondosum. The villi at the abembryonic pole degenerate. The chorion at the abembryonic pole looks smooth, and hence it is termed as chorion laeve. If the chorionic tissue was a contribution from the fetal side for the formation of placenta, from the maternal side the tissue contributing there is the decidua. It is the functional layer of the endometrium which gets shed off during parturition, and this functional layer of endometrium changes into decidua. How does decidua form? The decidua is formed as a result of hypertrophy and hyperplasia of the uterine endometrium stratum functionalis, in which the cells of the epithelium as well as the stroma synthesize a lot of glycogen, store within themselves, produce secretions, get edematous, and now they are called as, or the area, stratum functionalis area is called decidua. Depending on the region, the decidua can be termed as decidua basalis, parietalis or capsularis. Decidua basalis, also called decidual plate, lies over the chorion frondosum. It consists of a compact layer of decidual cells which are tightly connected to the chorion. The decidua capsularis is a decidual layer over the abembryonic pole. It becomes stretched and disintegrates with the growth of the chorionic vesicle. The decidua parietalis, on the other hand, is the rest of the decidua of the uterus. Initially, it is not in contact with the chorionic vesicle, but later it does come in contact with the chorionic vesicle. As the fetus enlarges, chorion laeve comes in contact with decidua parietalis and fuses with it, and thus the uterine lumen gets obliterated. The amnion and chorion fuse to form the amniochorionic membrane. Chorionic cavity is obliterated, and these membranes rupture during labor. With this background, we are now ready for the definition. Let us say it. Placenta is a hemochorial, discoid, villous, deciduate, labyrinthine feto-maternal organ. Let's break down the definition. Placenta is hemochorial. Hemochorial meaning that the interface in which the fetal and maternal tissues are coming together for the sake of exchange of nutrients and other products is, from the fetal side it is the chorion, and from the maternal side it is the maternal blood. Second word, discoid. The shape of placenta is like a disc. The placental surface texture wise consists of villi, so the word used is villous. The placenta is not a permanent structure, it's a temporary structure, and it is going to be shed off during the parturition. So the word added is deciduate, and the kind of network the villi and the intervillous spaces generate among themselves is a labyrinth. So the complete definition reads, placenta is a hemochorial, discoid, villous, deciduate, labyrinthine feto-maternal organ. The chorion frondosum is the only part of the chorion participating in the exchange of nutrients. The chorion frondosum with decidua basalis actually forms the placenta. Let's see the details of structure of placenta. By the 4th month, the placenta has two components, a fetal component and a maternal component. The fetal component is chorion frondosum, whereas the maternal component is decidua basalis. The decidual septa grow into the intervillous spaces from the decidua. They do not quite reach the chorionic plate. These septae divide the placenta into compartments or cotyledons. Each of this is a maternal cotyledon. Thus, between chorionic and decidual plates are chorionic villi, intervillous spaces and decidual septa. There are about 15 to 20 maternal cotyledons. Viewed from the maternal side, the cotyledons look like convex areas bounded by grooves. Each maternal cotyledon has a number of anchoring villi and their branches. One such villus and its branches constitute a fetal cotyledon. About 60 to 100 of those are present in fully formed placenta. At full term, on an average, the diameter of placenta is 15 to 25 centimeters. The thickness is approximately 3 centimeters and it weighs about 500 to 600 grams. After childbirth, the placenta is shed along with decidua. The fetal surface or the chorionic plate is smooth and lined by the amnion. Umbilical cord is attached to the fetal surface. Chorionic vessels converge toward the umbilical cord. So how does circulation of blood work in placenta? Well, maternal blood is brought to the intervillous spaces via the spiral arteries, and the venous blood present in the intervillous spaces is drained back by the endometrial veins. At the same place, the intervillous space, another set of vessels are present that are in the chorionic villi. There is a exchange of substances, nutrients, gases and other products between the intervillous spaces blood and the blood present in the chorionic villi. And after the exchange, the blood that is now enriched with nutrients and is devoid of those metabolic waste products present in the villi, the blood present in these chorionic villi will eventually be circulated throughout the fetal body by way of fetal circulation, and again will be brought to the same site for the process to be repeated over and over again. So we have umbilical arteries and umbilical veins in the umbilical cord. The umbilical arteries carry deoxygenated blood. The umbilical veins carry oxygenated blood. The umbilical arteries are bringing deoxygenated blood from the fetal body towards the placenta, whereas the umbilical veins carry oxygenated blood from placenta towards the fetal body. So what are the functions of placenta? Well, exchange of metabolic and gaseous products, transmission of maternal antibodies, production of hormones. These are some of the functions. In addition to that, placenta is also a respiratory organ, an excretory organ, and also a supporting organ. The normal site of formation of placenta is in the upper uterine segment. But sometimes the placenta might form below the developing fetus, that is towards the lower uterine segment. In such cases, that condition is called placenta previa. Sometimes the main placental mass can have an accessory lobe. In such a case, we call it placenta subcentriata. Another interesting way of formation of placenta is if the placenta is thin and is much more expanded. Such a placenta is called placenta membranacea. These are some of the clinical correlates of the placenta. Happy learning.